and Peng will be able to respond to them at that time. So our next speaker is Elizabeth Bess, and Elizabeth came to the center from Professor Matt Sigmund's group, and really bringing in sort of new techniques in terms of thinking about catalysis, in terms of bringing in new quantitative tools to analyze reactions. And again, she was very much at the sort of forefront of going further than where people had been before. And if I remember rightly, I think you spent some time at a pharmaceutical company as part of that sort of collaborative thinking, not just with academics, but also in an industrial setting as well, and made a big impact in that. And so as she was already going beyond the normal areas that most people would be thinking, it's not too surprising in their independent career that she's gone even further. And so her program now is a really interesting mix. We're thinking about fusing chemistry and microbiology together to understand chemical mechanisms by which the microbiome impacts human health and disease. And so you've got a lovely title. We're looking forward to hearing your talk, Finding a Catalyst of Gut Reactions. Welcome.
Thank you, Hugh. Let me share my slides here. Okay. Thank you for the opportunity to present here. It's such an honor to be back with this group, and my time as part of the CCHF now 6 or 7 years ago was really formative and actually helping guide my career to microbiology, and I'll tell a little bit of that story. So I guess the first thing to tell you is that you're not in the wrong spot. We are going to be talking about a little bit of CH functionalization, also the gut microbiome, and I'll tell you how my time at the CCHF kind of informed this transition. And it really comes down to, I think, this the idea that the CCHF is, I think, really promoting the beauty and joy in both collaboration, but also like cross-disciplinary discovery. And I think that can sometimes be daunting. And one of the things that was of such value to me in my time with this group was that it helped to break down some of those barriers of sort of diving into reactions or areas of science that you have no familiarity with, and then it's okay. And so that was something that was super valuable to me as my career went towards microbiology, and there were also specific interactions within the CCHF that kind of guided that. And I just wanted to highlight first here some of the folks that I had the pleasure of directly working with in the CCHF. And two publications that came out of those collaborations. One was a collaboration with the Dubois and Sigmund labs, where we were examining how we could use math to be able to pull out underlying features of molecules that we could use to predict selectivity of CH amination. With a collaboration at the Davies and Sigmund labs, we used a similar approach again to try to find what are these underlying trends, features of molecules that can help us understand patterns in reactivity. And with both of these collaborations, it exposed me to new ways of thinking, new kinds of metals that I hadn't worked with before, and was a real pleasure. I also had the great opportunity to go, as you mentioned, to Novartis in Basel through the CCHF. This was something that was facilitated through the CCHF, through Larry Hammond, and also worked with Stephen Lutz at Novartis Bioreactions Group, Vlada Urlacher at the University of Dusseldorf. And here we were thinking about how we could take what we were learning about developing models in CH functionalization settings, as well as in other settings, and apply them to better understand reactions that enzymes are performing. And it was through this work in Basel that I first got to touch and work with bacteria, which was such a joy for me, and enjoyed it so much that my postdoc became that. And I think that those opportunities in sort of growing up in science around these ideas of celebrating both collaboration and interdisciplinarity really helped to facilitate and smooth that transition. And the rest of my talk, I'll tell you about some of the chemistry that happens inside of you in the collection of bacteria that live in your GI tract called the gut microbiota. These are trillions of bacteria that live inside of you. And every person's gut microbiota is different. It's as unique as a fingerprint. There are similarities, but also substantial differences across people. And this variability is important because it has functional implications. So in three areas of many where the gut microbiota composition impacts health is energy harvest, drug efficacy, and disease risk. So if two people eat the same meal, they have two different microbiomes, each one of them can extract different amounts of energy from the same meal. Depending on which bacteria are in your gut, it also impacts the efficacy of drugs that we use to treat disease. So there are some bacteria that are able to metabolize drugs, making them no longer active, and in some cases, even making these drugs toxic. And the microbiome is emerging as playing an important role in this equation. Added to, of course, the role that human enzymes play also in modulating drug efficacy. The composition of the gut microbiome is also important in modifying disease risk. So there is an accumulating body of evidence that indicates that which bacteria are in your gut can predispose a person to disease or even protect them from disease. So in my lab at UC Irvine, we're really interested in understanding these processes at a mechanistic level. We want to understand how are bacteria interacting with their human host and what is the communication that's happening between the bacteria and its host. And we think that something that is key to this is molecules, the molecules that are trafficking in this space. So I introduced that the microbiota is the trillions of bacteria in your gut. The microbiome is the sphere in which those bacteria operate. So this includes molecules, proteins, metals that are circulating within the gut microbiome that are impacting the behavior of those bacteria. Also, these bacteria are doing many different kinds of chemical reactions. And there are huge questions that remain open in the field about which bacteria are doing what kind of chemistry. In many cases, we don't know what the substrates of bacterial chemistry are, what the products are, what enzymes are involved. And so there is great opportunity in this field to really dive into the chemistry that bacteria are doing inside of us that I think are an important aspect of how the microbiome is impacting host health. So what I'd like to do today is to describe a approach to identify the catalysts that are within the human gut that are doing chemistry on molecules that are abundant within the gut and how that can be leveraged to help us understand and maybe even engineer this space. And the case study that we'll dive into has to do with these two molecules that if you have eaten basically anything that has grown out of the ground. You have these molecules inside of you right now. These are pinoresinol and luriceresinol. These are molecules that are fairly abundant in the diet. And when these molecules, which are part of a class of molecules called lignans, when they reach your GI tract, come into contact with bacteria there, they get chemically metabolized to a downstream product called enterolactone. And enterolactone is a phytoestrogen that can interact with estrogen receptors. And through that interaction, there are several studies that show that when there are increasing levels of enterolactone within a human, there's decreased risk for breast cancer incidence. When rodent models of breast cancer are dosed with enterolactone, there's also a substantial decrease in tumor burden. What I became interested in is, how is this happening? What are the bacteria that are responsible? What are the genes and the enzymes that are responsible? If we know these kinds of things, we may be able to ask questions about if a person may be likely to benefit from eating a lignin-rich diet. We could use this information as predictive biomarkers. Also, if we understand how bacteria are doing these kinds of chemistry, it opens up opportunities for engineering the gut microbiota to promote beneficial chemistry. So what is the actual chemistry that's happening here? First off, when you eat pinoresinol, it's particularly abundant in seeds and grains. This molecule, when it reaches your gut, will interact with a bacterium called Egerthella lenta. About 2/3 of us have Egerthella lenta in our guts, and this organism will first reduce the oxygen-carbon bond at the benzoic position of pinoresinol and luriceresinol to form SACO. SACO then gets handed off to another bacterium in the gut that also most of us have, called Blaudia producta, and this organism demethylates SACO to DM-SACO. The next step in this pathway is a third bacterium that dehydroxylates DM-SACO at the 4 position of the catechol. And this product is handed off to the last bacterium in the process, Lactonifactor longoviformis. And this organism does oxidative cyclization to form enterolactone. These last 2 metabolites in this pathway are the ones that are considered bioactive metabolites. They enter the circulatory system, where they can have impacts on hormone-dependent disease, including breast cancer as well as prostate cancer. So what I was interested in is how we know these bacteria are doing this chemistry, but what is the source? What is the enzyme that's involved? And so we're going to go on a hunt here for what we'll call the benzyl ether reductase. We wanted to find the enzyme that we named benzyl ether reductase that would be responsible for doing this reduction of that benzyl ether bond in the first step of this pathway. And then what we learned from that first step, we applied to the other steps of the pathway to map what was happening here. So first, to dive in, we first took pinoresinol, incubated it with Egerthella lenta. These incubations are done in an anaerobic environment in a glove box, although there is water in this glove box. And these bacteria are able to rapidly metabolize pinoresinol to SACO in a matter of 2 to 4 hours. We then took a larger collection of bacterial strains of E. lenta and its close relatives. So here is a list of 25 or so strains of E. lenta, which means all of these organisms are the same species. Most of their genome is identical, but some of the genes within these organisms are different. And we incubated each of these strains with pinoresinol and found that 2/3 of these strains were able to metabolize the substrate. 1/3 of them could not. And what this difference in metabolism across strains allowed us to do was to perform comparative genomics. And what comparative genomics does is to basically take all of the genomes for the metabolizers and line up all the genomes for for the non-metabolizers, and then identify which genes are uniquely present in metabolizers and uniquely absent in non-metabolizers. And those are potential candidate genes for this chemistry. So doing this, we found only 2 genes out of the thousands of genes that are present in an Elenta genome. There were only 2 genes that were only present in metabolizers and not in non-metabolizers. One of these genes was a transcriptional regulator, so not involved in chemistry. The other one had homology to enzyme-like proteins. And so we took that gene, cloned it into E. coli to ask if we put this gene into an organism that is not able to metabolize penoresinol, does it give that organism the ability to metabolize this compound? And what we found is yes. When we put what we'll call this bare gene into E. coli, this organism was now able to convert penoresinol into SACO, indicating that this gene was sufficient for metabolism. So collecting all of those genomes from a panel of gut bacteria is a rather labor-intensive process. We wondered if there was a more direct route to get at this same information that we could use to better understand the other steps of this 4-component pathway as well. And what we turned to was RNA sequencing. So in RNA sequencing, we are sequencing all of the mRNA transcripts that are produced when in an organism. So when DNA is transcribed to RNA, then translated to protein, we can sequence those mRNA transcripts to see what genes are turning on in the presence of various stimuli. So we took Elenta, incubated it with penoresinol, and then sequenced all of the mRNA that was present as a consequence of penoresinol. You're seeing the results of that sequencing here. So in this plot is represented all of the mRNA in Elenta, and on the y-axis is fold change in how much RNA is there in the presence of penoresinol relative to the absence. And we were quite delighted to see this transcript here that is very distinct from the rest of the transcripts in this population, was upregulated several thousand fold over the bacteria in the absence of the compound. And it turns out that this transcript was for Bayer. So we have this strategy that we can use to identify genes that are linked to chemistry by being uniquely upregulated in the presence of their substrate. This kind of transcriptional profiling was performed to investigate the other steps in this pathway, the demethylation, dehydroxylation, and oxidative cyclization. And in each of these cases, we found genes that were upregulated that had unique signatures of enzymes that could be homologs for the chemistry of interest. And we have functionally confirmed the activity of EDL for this last step, and the other 2 organisms, we're currently in the process of confirming their activity, although based on their homology to known enzymes with known function, they seem like they are excellent candidates for being able to demethylate and dehydroxylate these substrates respectively. What we then went on to do was to take each one of these genes that is linked to the chemistry in this pathway and ask how abundant are these genes across people. So we took fecal samples from healthy free-living individuals and through qPCR measured the presence of each one of these genes across that population of people. And what we find is that about 2/3 of people are able to or at least have the entire pathway in terms of the genes that are linked to conversion of penoresinol to enterolactone. Another 1/3 or so of people do not have that complete set of genes, but most people do. This is interesting to us because not all people are able to equally convert penoresinol to enterolactone given the same amount of enterolactone, suggesting that there is an important component in this pathway of how efficient this pathway is. So what we think is important is how close these organisms are is key for their catalytic efficiency. And this is an idea that we are thinking about in the lab, and that many of the people in the field are thinking about, is how can we sculpt the chemistry of the microbiome in order to actually get the chemistry to occur that we want to occur when we want it to occur. How can we control some of these processes? One of the keys to this is thinking about what is the scope of molecules that can be metabolized by gut bacteria. A technician in my lab, Paola Bernardino, took a panel of plant-derived catechol and worked in collaboration with Vayu Rekdal from the Bolska's lab, incubating each of these catechol with that panel of Elentis strains that we saw a few slides back to identify what is the scope of dehydroxylation of these substrates at the 4 position of these catechol that can be metabolized. And what we find is that many of these substrates can be metabolized. In the darker the color, the more metabolism has occurred. And we see that many of these substrates are metabolized by many of the bacteria in the population. There are also numerous host-derived catechol, like dopamine, for instance, that also get metabolized by many of the bacteria in this collection. Although there are also substrates like epinephrine that are not very abundant in the gut that don't tend to get metabolized by these organisms. When we look at drugs and siderophores that also contain catechol motifs, there's much less metabolism of these compounds. And this suggests to us that the bacteria are evolving to metabolize most efficiently and effectively the substrates that are most abundant in the context of the gut, which are some of these host-derived catechols and plant-derived catechols. Current areas of gut microbiome research in my lab are to examine the role of gut bacteria in the gut-brain axis, which is how gut bacteria impact functions of the brain. We're also examining molecular level mechanisms by which gut bacteria interact with dietary fiber and metabolize and inactivate drugs. Just for a brief teaser of something to come from the lab, come out of the lab in the next month or so. We're investigating the role of the gut microbiome in Parkinson's disease. Parkinson's disease is a disease that is characterized by motor dysfunction that occurs when neurons in the motor cortex of the brain die. And what we have been examining is why and how. What are the processes in the gut that are leading to this? There's several pieces of evidence that suggest that Parkinson's is actually originating in the gut, not in the brain. So a phenomenal postdoc in the lab, Dr. Ortiz, has been examining this process, and the details will be forthcoming, but what I wanted to share here is that we've been taking germ-free C. elegans that are models of Parkinson's disease and colonizing them with bacteria in different ways and seeing some really fascinating phenotypes. So here you're seeing a video, hopefully this is coming through okay, of a worm that is moving quickly. This is a worm happy with life, moving with typical motor function. This worm is barely moving. This is Parkinsonian-like motor dysfunction. And the only difference between these worms is what is in their gut microbiome. And we're characterizing what are the specific molecular signatures of microbiomes that are responsible for these really dramatic differences in phenotype that we think are important for understanding Parkinson's. So stay tuned for more details there. Here's a throwback picture from the Sigmund lab, and Turnbaugh lab is where the pinoresinol work was performed. And here is a shout out to the folks in my lab who are making it so much fun to be in this job and to answer some exciting questions, I think, at the intersection of chemistry and the microbiome. And if you're ever in the Southern California area and you want to donate to science, we're always looking for donation of stool samples to be involved in research. So if you ever want to stop by and be involved in the science, we'd be happy to have you.
Well, that's a very interesting final comment. Yeah, lovely talk, Elizabeth. Really great. And again, there's questions coming in, and just for the sake of timing, I would suggest let's answer the questions in the chat and keep the discussion going there. So once again, let's join me and thank Elizabeth for a really interesting talk. You know, definitely moving into new areas beyond where she started, and also making it very accessible for us to follow. So thank you very much. It's great. Okay, so our last speaker is